going to talk about recent advances in genetics and aquaculture, but particularly from the point of view of world fish and the work that's involved in that organization. Now, for those of you who don't know, World Fish is one of the 15 or so CGIAR centers whose headquarters are spread in various parts of the world. But you'll see that World Fish's headquarters is in Penang, in Malaysia. And our activity is spread through a number of countries, but focused mainly in South Asia and Sub-Saharan Africa. And why fish? Well, fish is important because 16% of the animal protein in the diet of people worldwide is from fish. And in many places, particularly Africa and Asia, it's 20 to 30%. And nowadays, some 50% of the fish that people eat in the world comes from aquaculture. But if we look at the domestication of species in aquaculture, we can see a very interesting parallel, first described by Duarte et al. About 15 years ago. And you can see on the left, the land animals were domesticated around 10,000 years ago. And then there was a very rapid domestication through up until about 5,000 years and tailed off a bit, but extremely rapid through that time period. But for animals, although around about maybe 2,000 years ago the Chinese were working with carp, it really exploded for aquaculture starting only about 70 or 80 years ago, but there's been a tremendous number of species domesticated since then, or at least worked with. So, 400 species cultured, 90 domesticated, by which I mean there's 3 or more generations in captivity, but not necessarily subjected to genetic selection. And within those, only about 5% have significant genetic improvement programs. And if we look at what happens in animal and plant breeding, I don't want to go into the detail of this, but I just want to indicate to you that in plant breeding, it takes about 10 years from the start of a particular improvement before that actually gets into farming systems. There's a set of procedures of population improvement in the first couple of years, and as they move through the process, they get into further product development. But the critical part here is at the end, there's somewhere between less than 100 or less than 10,000 genetically different individuals in the population that is released. And that means that people very often think of looking at crops, but there's a limited genetic diversity in what is released in a given year, which is true to some extent. But if you look at animal breeding, you can see it all occurs in a population of organisms. Instead of taking a few genotypes and expanding their number through this step, so you end up with a smaller number of genotypes produced, this is an activity where you've got a population with a very large number of genes, assuming you start with a big enough population, and you can then end up with something like often more than 100 million genetically different individuals, depending on the overall size of your population. So very different processes. So if we now look at a brief history of a recent history, so not the 10,000 years ago where people started to domesticate animals, but after they rediscovered the work of genetics and inheritance, which was about the late 1800s, where that was rediscovered. And in the boxes I've put some of the key components that are used today. So population genetics was introduced as an extension of the laws of inheritance. So Mendel discovered the laws of inheritance back in the late 1800s. This became developed as a much bigger theory, which then became extended in the 1950s into working at the specific breeding values of individuals. Then that became computerized with blood programs, providing even more precise estimations of breeding value. And then today we've got molecular markers used to do that. Now, in parallel with this development of the statistics and analysis of relationships of organisms was the development of molecular information, starting in 1950 with the model 53, with the model of DNA structure, then critically 20 years later working with genetic engineering and the first recombinant DNA molecules. This led to the development of a range of biotechnologies in the early 80s, and then critically, these two sets of activity fused to get molecular markers which could be used to further improve the accuracy of selection. And throughout this period, we begin to see in the 80s also the development of ICT and computers, which speeded the whole process of calculation of statistics. It speeded the whole process of the calculation and understanding of molecular events, including protein-protein interactions, being able to generate more information from chemistry and so on. And today, we then have the introduction through this process in the boxes of genomic selection when fused with molecular markers, which again has increased the speed and accuracy of genetic improvement. And then ultimately 2013, now 10 years ago, the introduction of CRISPR-Cas9 based genome editing. And so in the next part of the talk, I'll talk a little bit more about each one. But you can see in the domestication of animals and plants starting 9,000 years ago, since 1920 and the introduction of an understanding of genetics to those systems, the speed of genetic improvement has increased immensely. And central to that is this concept of selective breeding. And selective breeding at one simplistic level is that you take the extreme of the distribution of a, for example, size, in this case length, and you can see if you take the longer ones, the bigger ones, and you breed them together, the next generation you've got a population on average that's bigger, and you keep repeating that process until you get an improved organism for the characteristic that you're selecting. Now this in a sense is what people did 10,000 years ago, but they chose animals that look big. But the issue here is that depending which environment you took from them, a small animal that grew up in an environment that was not particularly nutritious would actually have the genes for bigger growth if it was in the correct environment. What happened when people understood genetics more was that they realized that they had to choose the animals that had the genes to make themselves big, and they weren't big just because they grew up in a better environment than the other individual. And to that end, they started working with pedigrees, and here on the left you can see an example of about 34 generations of a species with all the males and the females tracked, and you knew the relationship of each individual in that pedigree. That allowed you to be able to predict more clearly the breeding value of any individual because you knew it was carrying genes that would make it true breeding for that particular characteristic. And of course, as the populations get bigger and perhaps you deal with more characteristics, it can get really complicated on the right-hand side. And I defy anybody to sit with a paper and pencil and work that one out, which is why the advent of computers and automated calculation made such a huge difference to the ability to deal with much more complicated circumstances. So coming back to this, if we look at a particular advantage of fish, we can see in the normal structure for a breeding program for animals, you've got a nucleus breeding center, which is a relatively small population, and you do your selection within that small population. When I say small, it has to be somewhere really about a minimum between 100 to 200 breeding pairs. And the issue here is that the smaller the population you have, the faster the genetic gain that you can have from this controlled mating of pedigreed individuals. But the smaller the number of the population that is involved, the greater the rate of inbreeding, which leads to deleterious effects in development and ultimately to poor growth. So it's a balance between the two. It's not impossible, but very difficult to work with 1 million individuals. So you work with 200. But if you only work with 10, yes, the gain immediately will be very quick, but the inbreeding will catch up and make a mess of things for you. But the crucial issue I want to indicate here is that in agriculture today, the gain each generation is only about 2%, sometimes far less than 2%, whereas in fish to date, what we're seeing is somewhere in excess of 8% for almost every species that's being worked with. The other critical issue is the number of young. Cattle, sheep and others only produce a relatively small number, pigs a bit more, chickens a bit more. But when you compare that with fish produce anywhere from 1,000 to 1 million eggs, and therefore the ability to be able to multiply your improved stock is immensely greater. So the opportunity to start from zero and produce improved fish and get them into a farming system because you've multiplied the number of improved individuals needed is actually way bigger than in agriculture. And one can actually look at taking a 10-year period starting from scratch to be able to produce an improved fish that's actually productive within a farming system. And that's a very realistic prospect. In addition, and then we've seen just in the last 20 years, for me, recent is a bit longer than it might be for some of you, but here it involves using a reference population of fish where you know the characteristic, you've made all the hard work of measuring it, and then you measure their genotype using tools that are able to identify the different molecular types in the fish. And it's denoted here by these bright blue dots which indicate the fish have that particular genetic variant. And then on section 2 you can see that you can calculate what's called the genetic breeding value, or the genomic breeding value, sorry, by taking all these molecular markers and associating them with the actual measurement of the phenotype. Okay. From that, you can then screen a whole lot of fish, so you increase the population size you work with, and you then select the breeders from that. So it's a way of multiplying the population you're working with to then select far more accurately the fish that you will breed from. And then finally, in the last decade or so, that's CRISPR-Cas9. So basically gene editing, which uses a number of tools. I'm not going to go into the detail of this, but essentially they're molecules which will snip the DNA at a particular point. There's 3 or 4 different tools listed on the top of this slide: meganucleases, zinc finger nucleases, TALEN, and CRISPR-Cas9. They're all molecules that will take a piece of DNA and snip it. And they'll be able to do that in a very, very specified fashion. And ultimately they then take that and join it again. So they can be used to remove a piece of DNA, they can be used to change a piece of DNA, they can be used to add in a piece of DNA, and it's gene editing. So very specific genetic changes to particular genes. Now this is different to GMOs in the sense that GMOs also snip DNA and put things in, but they also leave a particular segment of DNA which legally classifies them as a GMO. But to do that, you've got to use gene editing in an early phase usually the single cell phase in a fish, or any organism actually, and that needs to take place that early because you want that change to be in every cell in the organism's body as it grows up. So when it matures, every cell has that change so that it can be heritable. And that itself is a more difficult component sometimes than actually making the gene edit in the single cell. And there's examples of that, for example, in the lower set of photographs, deletions in red sea green from a myostatin gene gave fish that were 16% in more muscle mass and then a relatively smaller, shorter body length. And you can see those bigger fish on the left. Now, that's also been achieved now in a whole range of fish: common carp, olive flounder, black snake, green, sea green, salmon, a whole range of species. So this is a very, very specific technique that allows one to change particular genes that are able to have a highly significant effect on a particular physiological pathway. And to date, people have normally focused on growth. They've focused on getting more muscle mass, which is the flesh you can eat. But it could also be related to disease resistance and other characteristics. And that includes color change in tilapia. So the fish on the left has been gene edited so that it's lost pigment, and presumably because it's paler, is more acceptable to customers in the market, assuming that they are comfortable with gene editing. And as I said, this then could potentially change a whole lot of things like growth. And here's the big one: so-called complex traits that are important for the resilience of the fish. Disease resistance I've mentioned, but others are being able to continue to grow in stressed environments. Furthermore, to control their reproduction, and that becomes important because people are concerned about the introduction of high-performing fish into new environments, and so the escapees they're concerned will interact with wild populations and others. But if you could cut off the reproduction, and that's now being done by workers in Norway who are working with a gene called the dead end gene, which means that the cells in the gonads don't perform and die, which means that the fish are not reproductive, which means you can have production populations which are non-reproductive. The problem is you need to work out how to keep your breeding programs going. So it's not just that particular element, you've got to look at the whole production process. And so the issue here is that these technologies in combination can be used to protect biodiversity, potentially by being able to control reproduction of domesticated populations, and potentially could also be used to enable more species to be developed for culture. Because one of the key issues in agriculture, but it's also absolutely clear that it's something affecting aquaculture, is there are relatively few species which are domesticable. And in agriculture, it means that ultimately there's 4 or 5 species which are responsible for something like 80 to 90% of the production of the food that we eat. And that process is occurring similarly in aquaculture, where there's about 10 or 20 species that are responsible for 80% of the aquatic foods that we consume. And that's because they can be cultured easily, they have the growth and characteristics that we want in the food that we want to eat. And because they can be cultured, because there's something that provides us with nutritious food, that's the ones that are being developed. But now there's a call for greater biodiversity in our foods. But many of those species don't grow nearly as well as these other ones, and the consequence is that if we wanted to grow them, we'd have to use far greater land areas in the case of terrestrial agriculture, or far greater areas of water, and far more food be able to develop those species than those fast-growing ones that we can farm easily and people want to eat. So you're beginning to see that there is a whole range of trade-offs at every instance here. Do we want a more biodiverse agriculture, a more biodiverse aquaculture? If we do work with a range of species, how do we get the performance up so we can actually have an efficient food production system? And in addition to that, if you do modify a farm to create a range of species, they will interact more with others in their native areas than perhaps the strains that are introduced. But that might be an issue that could be countered by controlling reproduction, which might be best easily done by gene editing. But then we're talking about a technology, perhaps which people are not so comfortable with. And the issue as to why we need to be really efficient in our food production is simple. There is still an increasing global demand for food as the human population is still growing. So we need more efficient food production from that perspective. We also need to have the food being produced by organisms that are under environmental stress, partly because of climate change. So we need to help them adapt to climate and keep producing. I've already talked about the biodiversity management. And we need all of those things for people to have profitable circumstances, for them to have livelihoods and therefore a sustainable food production system. And the example here on the left from Scotland with all these red dots, which are fish farms, shows you it's all on the west coast, and that's because the environment in that area is sufficient to allow sea cages to be worked with. The open coastlines on the east coast simply wouldn't sustain those. They're too exposed to weather. And in addition, if you look at that top photograph, which is the Mediterranean, you'll see the light blue areas indicating conservation zones. But equally, there's a whole set of other uses for the marine environment. So the areas that one can use to farm aquatic foods are limited, and that's another reason why we have to have much more efficient processes to produce them. So we've seen tremendous improvements in salmon, in white shrimp, and in tilapia, and that's made an important penetration of the market. But for many production systems in aquaculture, the penetration of improved strains is very low, around 12 to 15%. Sometimes that's because there is no improved strain available, but in other cases there's a whole range of reasons why they may not be taken up. But it so concerned the FAO that in its first State of the World's Aquatic Genetic Resources published in 2019, their subsequent global plan of action included the need to accelerate the use of genetically improved material in aquaculture. And why is it that there isn't so great an uptake? Well, the reason is it's not all because of genetics. There are key factors involved in whether a particular strain is adopted. The cost of the strain. Does the improved strain meet the needs of users? Does it meet their perceived needs, even if it meets the needs that are actually there? Is it something that they see as valuable to them? There needs to be enabling policies for the development of an aquaculture industry and to allow farmers to work effectively. There needs to be effective dissemination systems for seed, but also sound management practices and the ability of farmers to access key inputs that are necessary for them to grow their product, feeds, healthcare, as well as quality seed. And there needs to be effective access to markets. And they also need to have a commercial capability of some form, at some level. If you don't make money at it, then you're losing money, and so it will not be sustainable. So there's a whole range of factors that need to be included before the technical developments within an improved genetics is going to be resulting in an effective change in a farming system. So where has World Fish Actions been in this area? And this is where I'm going to look at some of the recent improvements in genetics in world fish. World Fish was responsible about 20 years ago for the first improvement within tilapia, genetically improved farm tilapia, GIF. It grows up to 50 to 80% faster than other strains. It has a high survival. It's been distributed now widely in Asia, South America, less so in Africa. In the Philippines, it's more than 70% of the market, and GIF technology is used in many other regions. Our estimate, and it's a rough estimate because it's very difficult to get good data, our estimate is that about 50% of the global production of tilapia today, some 5 or 6,000 million tons, is about 50% from gift or gift-derived germplasm. That's a huge impact worldwide. Now that was focused on a particular trait of growth, and we've been working on new traits to extend the resilience of gift tilapia. And you can see here the growth over the last 40 years in tilapia and aquaculture. The vast majority is non-tilapia, and the vast majority of that material is coming from genetically improved programs. So you can see the potential scale of the impact. What we've done since then is in the last 7 years, we've developed programs for 3 species of carp in Bangladesh: rohu carp, katla carp, and silver carp. And we've set up base populations of more than 150 breeding pairs. We've got a range of molecular markers developed for them. And each of these fish is growing together in polyculture ponds. So we're beginning to develop a key technology for one of the most productive aquatic farming systems in the world in Bangladesh. And in relation to that, we're starting work on African catfish. And here you can see the early stages, which we do African catfish, are assessing the traits that people need, first and foremost. What do we improve? Developing the enabling technologies. How do we actually get things that work to husband the fish and so on? Then in the intermediate stage, which is where I say our carps would be, it's improving growth strains are available, and we're starting to assess them on farm performance. And I can say to you now that the rohu carp that I showed you in the previous slide, it looks like after 6 years, because it's 3 generations, 2 years per generation, that we've got fish that are 30% faster growing than the other strains. Now you imagine if we could actually get those fish taken up by the farmers, and that adoption could result in something like a 20 to 30% improvement in fish, or greater growth in fish production in Bangladesh. Then finally, working on more complex resilience traits, which are technically more difficult to do, but incredibly important to have developed. So we've got fish that survive well, are able to continue to produce under stress, and particular stressors that I'll talk about later. And in that regard, we've now got a series of products that will emerge through 2024 to 2030, and they're listed in that table there. So designing an African catfish program in 2024 with an aim by 2030 to have that regularly released in sub-Saharan Africa. With the carps, rohu's being released this year, we're assessing it. We aim to release silver carp by 2027 and improved cattle release by 2030. All based at the moment largely on selective breeding, that very important process that I've described earlier. With the tilapia, we've been working on 3 new resilience traits. One of them is resistance to tilapia lake virus, and that's aimed to be released in 2024, and then further more in the next set of 3 years. It will add further resilience traits to that, probably related to temperature, to the ability to work in or survive in waters at higher temperature and reduced oxygen. And then a regular release of those through to 2030. One of the key points I'm going to make is that each of these fish is relatively low in the food chain. They're not carnivores, although they can be, but they can all exist on, they either feed on plankton, phytoplankton, zooplankton, and certainly don't need to have fish oil in their diet to survive. Some of the additional growth they might want, or some of the reasons why fish oils are added towards the end of their period of production is more about the value of these fish as human nutrition as opposed to the need for those fish to have that in the diet. So they, and most importantly, collectively, these species are responsible for 28% of the finfish production worldwide. So if we are able to develop these technologies effectively, if we are able to improve the traits, this could be a tremendous impact globally, in addition to specific impact in some of our key target countries that we work with in Africa and South Asia. So we're starting analysis of traits, and here you can look at the comparison between female and male preferences, in this case in Bangladesh, and you can see while there's some minor differences, broadly it's similar for the traits that we tested. We'll be testing those traits with great further market segments for all the species we work with. So is it what our users want? We've started to use molecular analyses here, and just look at the diagram at the right hand side. Those pale boxes along that diagonal are signatures of little pockets of inbreeding within that population of fish that we assembled. And so we were able then to modify the pedigree we work with to remove that inbreeding, which means that we'll have a far healthier population going forward and a much more productive population as time goes on. We developed a tilapia SNP array, and for those of you who are involved in that process, it's essentially a means of identifying different DNA variants at particular pieces of the DNA repeatedly. And you can say then something, in this case, something around 65,000 different loci at one time. And you can do it repeatedly at different stages. And this tool is open access. You can get it through Axiom arrays. And they do ask us for permission to release it, but it's available. The only openly available SNP chip to date on tilapia. We've been able to use our work mainly also because we were affected by TILV here in Malaysia. TILV is a virus that causes high mortalities in tilapia, appeared about 3 or 4 years ago, gave rise to great concern. And we were able to show that there was a huge variation in resistance between different families. And in work with the University of Edinburgh at the Roslin Institute, we were able to determine that there was a high heritability for disease resistance. And we are now breeding for a strain that, like I said in a previous slide, we aim to release in 2024. We also have patents pending on biomarkers in relation to that particular disease. With the Earl Institute in Norwich, we've been working on the GIFT genome sequence, and if you can focus on the diagram in this bottom part with these little blue components, and forgive me because I'm red-green colorblind, I can't describe the color with the ones going down. But this is specifically been designed for GIFT, and it shows major differences with the publicly available Nile tilapia genome sequence. And what I'm showing you here is that it shows clear portions of the genome of GIFT that come from aureus genome, so the aureus species, which is cold adapted, And you see major blocks of that in this top component showing those blue peaks. Then you've got some very large peaks of the other color pointing down, which have come from Mozambique. Now, one of the things that we'll be looking to see is to work out whether these are events that occur a long time past in evolution, because many tilapia species interbreed in the wild, or whether they're actually more specific elements that have occurred recently. And this will provide a very strong basis for being able to understand the genes and to map those genes for use in elements such as gene editing and to support the selective breeding process. So going forward in terms of future work, we're working on those new strains which I've indicated in 4 different species, a greater range of species than we had in the past. We're developing new resilience traits which will be important for climate adaptation. We're working with latest technologies of genomic selection, and we're continuing to develop new tools with our partners on genome sequence, using that to develop more with functional genomics to get a clearer annotation of that genome sequence, which will allow us to identify the specific gene pathways that are important to understand to identify which genes might be the focus of gene editing. And all ultimately to enhance a global impact with respect to producing practical strains of fish that work effectively for them. To achieve this vision, we need to get new significant investment, which we are working on with industry partners and others, developing an internationally credible research team with partners in Europe, Asia, and in Malaysia. And through strong global industry partnerships. Again, the issue here is not just the technical developments, but our ability to be able to transfer those effectively into farming systems. And particularly through our new initiative in aquatic foods, we'll be using that to assess some key factors that are affecting all these other issues that I mentioned before that we need to address to be able to effectively and safely introduce these technologies and place them in the hands of the ultimate end users, to enable governments and farmers and investors to make a choice between whether they develop indigenous species, which may have characteristics that are far, far less immediately available for farming and strains, or how to engage in developing safe introductions of elite material. And all of this is with the aim, finally, of having a greater flexibility for farmers. In other words, a faster growing fish can be grown for the same period of time to get a bigger fish that might achieve a greater market price, or they could grow the fish for a smaller period of time to get the same size of fish, which means that they can actually get an income in a shorter time period. But the farmer can make that decision because he's got a fish that allows him to do both of those things. And similarly, if we could get a fish that's with far better survival, necessarily produce more, but they have anything from 40 to 60% higher profitability, which means that they have a sustainable livelihood. And again, reduced environmental impact. These improved strains produce more product, but use less water, use less feed, because one of the reasons why in Bangladesh GIF was actually much more profitable was that it was actually far more effective in its use of feed, which was a major cost of farming. So all of these things, fancy biotechnical approaches, fancy bioinformatic approaches, are all complex interventions that are designed to produce a product that is of use to the people who will ultimately use it and provide them with clear benefits that will also provide global benefits with respect to reduced environmental impact greater sustainability of food supply, a better climate adaptation, and therefore greater